my video presentation about food microbiology research portfolio. Uh, the title of my video is Food Microbiology Research Portfolio. The introduction. Food microbiology examines how bacteria, fungi, viruses, parasites, and prions, which interact with food, either as beneficial agent in fermentation and probiotics foods or as culprits of spoilage and disease. From lean work first observation to pasture breakthrough that field underprints modern food safety, food preservation technologies like pasteurization or refrigeration, and the development of functional food rich in probiotics. Section A. Section A talks about microorganism in food chain. Bacteria 1. Pathogens like Salmonella, E. coli 0157, and Listeria, which cause illness. Spoilage species, Pseudomonas degrade sensory quality. Beneficial lactic acid bacteria like Lactobacillus, Streptococcus, which drive fermentation. 2. Fungi. Fungi is example like Saccharomyces cerevisiae produce alcohol and carbon dioxide in baking or brewing. Molds, Penicillium, Aspergillus, which spoils food but also ripens yeast. Okay, 3 viruses. Norovirus and hepatitis A transmit via contaminated food, causing gastroenteritis and liver disease. Okay, prions. Prions which misfolds proteins resistant to standard sterilization. Example, BSC made cow agent require strict control in animal products. Okay. Parasites, protozoa, Gigelia, Cytopodrium, and helminths, Trichinella, Toxoplasma, which infect through uncooked meat or tinned products. Section B. Section B talks about Section B talks about source and routine of contamination. The farm to fork consume highlight critical contamination points. On farm, soil, water, manure, livestock, seedling, pathogen. Okay, harvesting and primary processing, poor hygienic, uh, unsterilized tools, contaminated wash water. Processing and packaging, cross contamination, temperature abuse, tinned packaging. Okay. Distribution and retail, break in cold chain, prolonged storage, customer handling, uncooked shared utensils, inadequate hand washing. Preventative measures include good agricultural practice, worker training, HACCP implementation, sanitation protocols, cold chain maintenance, and customer education. Okay, Section C. Section C talks about detection and control of microorganisms. Detection method, PCR for rapid sensitive DNA-based pathogen ID. Okay, ELISA for antigen antibody screening of toxins and microbes. Culture-based analysis for live cells enumeration. Okay, control strategies, sanitation of surface and equipments, personal hygiene and protocol control of handlers, HACCP to identify and manage critical control points, and temperature control to stay out of 5 degrees Celsius to 60 degrees Celsius danger zone through cooking to laterally target pathogens. Case study. Listeria monocytogenes in ready-to-eat food underscores the importance of environmental monitoring, cold chain integrity, and sanitation in protecting vulnerable population. Section D talks about microorganism in food fermentation. Fermentation harness beneficial microbes to enhance mm, prevention, flavor, and nutrition. Okay, one. Lactic acid bacteria, uh, lactobacillus in yogurt, kumsi, sauerkraut, sauerkrauts, yeast like Saccharomyces cerevisiae in bread, beer, wine. Okay, moles, penicillin, aspergillus in cheese, ripening, and soy fermentations. Okay, kumsi as a role as a model sequential lab activity acidifies vegetables, creating probiotics, vitamins, antioxidants, and antimicrobial condition control. Stata culture and hygienic practice ensure safety and consistent quality. Section E talks about emerging issue in food microbiology, antimicrobial resistant or AMR, overuse of antibiotics in agriculture, which select resistant strains in food and environments. Novel pathogens are new or re-emerging microbes like E. coli 0104H4, hepatitis C vitamins, which demand uptake risk assessment and diagnostics. Microplastics, and this practice subtle microbes and resistance gene through water, soil, and food, complicating contamination control and hygiene. Okay, as one health approach linked human, animals, and environmental health alongside genomic surveillance and alternative microbiomes is vital for future reliance. A reflective insight. Developing the portfolio video transformed a basic concern with contamination into a system level appreciation of microbial rules. From farm practice to consumer behavior, the balance of safety and innovation, especially via fermentation, emerged as central. Conforming AMR, emerging pathogens, and microplastics has highlighted the field dynamic challenges and ongoing need for research policy and interdisciplinary collaboration. And this brings to the end of my video.